Hi, everybody. Welcome to OsteoBites. Thanks so much for taking an hour out of your day to join us. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Amy LeBlanc from the National Cancer Institute. She'll provide updates on the NCI DOG2 project and discuss creating a translational genomic framework for canine osteosarcoma. Thanks so much, Dr. LeBlanc, for joining us on OsteoBites today. We are thrilled to have you, and thank you also to our panelist, Samantha, for joining us. A little bit more about our guest today before we get started. Dr. Amy LeBlanc is a board-certified veterinary oncologist, senior scientist, and the director of the intramural NCI's Comparative Oncology Program. And in this position, she conducts preclinical mouse and translational pet dog studies that are designed to inform the drug and imaging agent development path for human cancer patients, specifically those with osteosarcoma. She directly oversees the NCI Comparative Oncology Trials Consortium, which provides the infrastructure necessary to connect participating veterinary academic institutions with stakeholders in drug development to execute fit-for-purpose comparative clinical trials in novel therapeutics and imaging agents. Her program provides support to several extramural NCI-funded initiatives, including the Integrated Canine Data Commons and Cancer Moonshot-funded canine immunotherapeutic clinical trials conducted under the Precinct Network. So we're so excited for this talk today, Dr. LeBlanc. Just a couple of announcements before we get started. I wanted to highlight a resource that we have on the MIB Agents website that might be of particular interest to our audience today. We have a canine trials directory that lists some resources about canine clinical trials, including a list of veterinary oncology programs around the country so that caregivers can find a program near them, as well as a list of canine trials. And if you have an academic institution, if you're at an academic institution and you'd like to be included, just email me at christina@mibagents.org. And registration is filling up really quickly for our 2024 Factor Osteosarcoma Conference, which is June 20th to 22nd in Cleveland this year. We are definitely gonna fill, we're close to capacity already, and we've been trying to actually add some more spots to accommodate. So definitely register soon if you are thinking about coming. I'm gonna put a link in the chat where you can find information about our panels, our speakers, our various wellness sessions, and even the prom that we're gonna have this year on Friday night. We have amazing speakers, including Dr. LeBlanc is going to be there. And we have so much good scientific content. You will learn a lot. But I think really the most special thing about Factor is what you will feel when you're at Factor and when you leave Factor. You know, let's be real, like working in oncology is a really tough gig, especially in osteosarcoma when you're dealing with kids and doggies facing a really tough disease and sometimes with poor outcomes. So, you know, moving research along and to clinical translation can take a long time and it requires a lot of perseverance. So when you come to Factor, you hear about all the fantastic work and the strides that scientists are making and how there's so many people tackling the problem from different angles. And you'll meet patients and caregivers who are so hopeful for your work and whom your work is directly impacting. You will really leave re-energized and reinvigorated with a deep understanding of how impactful and meaningful your work is. And don't we all need that once in a while? So really hope to see you there. If you're on the fence, come. And also Samantha will be at Factor this year, which is yet another reason to come. Samantha, could you please introduce yourself?
Yes, so my name is Samantha Ulloa. I usually go by Sammy. I'm 21 years old. I was diagnosed with osteosarcoma at 19 in 2022. I underwent treatment for about a year and I had a limb salvage surgery, so I have an internal prosthetic in my right leg. And I'm about almost exactly a year NED since I rang my bell, so that's exciting. And I'll go ahead and hand it over to Dr. Amy.
Awesome. Thank you, guys. It's such a pleasure to be here. And I completely echo the sentiments about Factor. It is hands down my favorite meeting ever. Seriously. It's a completely fresh perspective for a scientist, and it's just an energizing, emotional, scientifically rich meeting. And I just love it. And I can't wait. So see you there.
Yeah.
Yay. Yeah. So let me just share my screen real quick and we will get started.
And everyone, please feel free to put any questions in the Q&A.
Oh yeah.
We'll likely hold those to the end because I know we have a lot of great content to get through today, but please feel free to add any questions to the Q&A.
And interrupt me if there are immediate questions about slide layout or if something's not clear. Happy to really keep this open and interactive. Does that look okay to you guys?
That looks perfect. Thank you.
Well, let's just jump in. Okay, so some themes for today. So I think the first thing that I want to convey is that pet dogs are cancer patients as well as maybe a cancer model, right? So as veterinarians, we think of them as our patients and we care for them at the highest level. So everything that you're going to see today that involves dogs, really think of them as, being cared for in a clinical setting that really strives to deliver the most high quality care, and think of them as patients and part of our families. I think all of us agree that osteo in humans and dogs, there's a lot of really interesting themes there that echo through comparative biology and unmet clinical needs. So we're going to talk about that. I'm going to introduce you to our program and the Dog Squared project and what we're trying to accomplish, and really also where we hope to go. So I've met some amazing people recently. I met Serena on the Hill. She is a patient with osteo that really, again, connected with me, and she came and visited the NIH and told us about her journey, and we continue to keep in touch with her. The MIB Osteo Warriors came to visit us at NIH recently, which was wonderful. And I, connect all these wonderful experiences also with meeting families that have dogs with osteosarcoma. So this is a dog named Dudley. He lived in Rockville for about 10 years, and he had an osteosarcoma in his distal radius. And we learned a lot from him and are continuing to learn from him, as I'll show you today. So I think all of us know about metastatic progression and how that really is the thing that we are fighting every day, because it is the hardest aspect. There are so many hard aspects of this disease, but this is where we need to focus our efforts, in my opinion. So dogs, they will develop metastatic disease, and unfortunately, they have a very short period with us, even with therapy, and a low percentage of long-term survival. And I think that studying dogs with osteosarcoma, because of the commonality between their disease and human disease, it really gives us an opportunity to learn and advance, because we haven't seen many improvements in outcome for a long time for both species, and we need better biomarkers to help stratify patients and personalize therapy. So one way that we're doing that was actually through, through the canine biospecimens, tumor samples that were harvested from dogs that enrolled in this large clinical trial. So several years ago, under the guidance of Chan Khanna, who I think probably many of you know, who started comparative oncology here at the NCI 20 years ago, he and Tim Fan and others that you may know on this author list, we all worked together to ask a question about adjuvant sirolimus, which is a drug that inhibits the mTOR pathway. And based on a lot of preclinical data, there was strong evidence to believe that there would be a survival outcome benefit in dogs. So we worked really hard with the Morris Animal Foundation support to conduct this large trial. And unfortunately, it was a negative trial. But we did learn a whole lot of really important lessons from this trial. We learned that we need to do a better job with our standard of care for dogs. We learned also that clinical trials like this are a great way to collect outcome-linked biospecimens for the future of biology in this disease. So when we embarked upon this project, we basically said, okay, we have all these samples from 324 dogs, and we've got funding support from Morris and from the NCI. What can we do with all of these samples? And so this just gives you sort of an overview of the types of samples we collected and the different methods we used to analyze them, because we wanted to create a framework for the disease that we could really deeply understand biology of primary tumors and metastases. We wanted to work towards generating new biomarkers that we could apply in the clinic at the time the dog is diagnosed with osteosarcoma to help us better understand what might happen in the future with treatment. And really, the more that we work on this project, the more we realize that we've got this 22,000 samples, I think, is outdated. I think it's actually more like 24 to 25,000 samples that really span all these different fluids and tissue types, and also data sets that really become quite valuable, not only to our group, but to others in the community. So this is just sort of a snapshot of some of the projects that have sort of blossomed right out of this effort. And we're working with people both internal to the NCI and NIH, but also folks out in the community, because we really feel like this is an opportunity for everybody to work together and, again, create a framework for the disease that we can use to understand dogs better, but then also just do a better job of relating the disease in dogs to the disease in people to really benefit everybody. So these are some of the projects. I'm going to do a little bit of a deep dive on some of them. And I guess a takeaway message here is that there's a lot going on, and there will be more updates to come in the near future. Okay. So overall, what's the goal, right? So create a genomic framework, translational framework that has value for both species, and really think about sort of layering all these data sets together to create biomarkers for outcome prediction and response assessment. So we're really thinking deeply about, patient factors. So, what breed of dog, where is the tumor located? What are its molecular features? What does it look like under the microscope? And can we use AI and machine learning approaches to extract features that may not be visible to the human pathologist eye? And what's the immune landscape look like? And I think all those things together are helping us get at, in the canine space, like, who are the high-risk patients? Who are those that may have a really short disease-free or progression-free survival, that really are the patients that are in the most dire need of improved outcomes? And understanding that group, then it makes it easier for us to understand all the other groups of patients for which we can do a better job tailoring therapy. So the first thing that we thought would be important to do was create a comparative pathology board. So this is essentially a group of pathologists that have a really strong interest in osteosarcoma pathology to get together and really, first and foremost, talk to each other. And when physician and veterinary pathologists talk to each other, really amazing things happen. We have the goals in mind to really compare and contrast what osteosarcoma looks like under the microscope. And working together, we can establish updated criteria for diagnosis in dogs with insight from the physician side of the equation. We can then layer on all of this molecular data we're generating. And when we do that, it really helps move the field forward. And we've done this a couple of other times with brain tumor boards. We did a canine glioma and canine meningioma board. And building upon the experience of those boards has really helped us get to an exciting place with this osteosarcoma board. So the pathologists on this slide, Courtney Schott, John Bush, and Jess Beck, who's in my group, are working with a large group of tumors. There's 200 dog tumors that have a scanned image of the tumor on a glass slide, along with data from immunohistochemistry, sequencing, spatial profiling, et cetera, and really have the opportunity to think not just about what the tumor looks like under the microscope, but what other molecular features might be associated with how it looks, and whether or not what they can characterize in the dog is also seen in human. So here's an example of one of the projects that can sort of, again, bloom out of a pathology board like this. So we have an artificial intelligence resource within the branch that my program belongs to here at the NCI. And one of the things that having an AI group lets us do is ask questions about AI extracted features in scanned images of tumors. So again, going back to our canine sort of biobank that we created from the big trials, we have about 300 scanned images of the canine tumors. And Sushant Patkar, who's a really talented postdoc in the AI group, worked with Jess to annotate the different regions of these tumors. And so what I'm showing you here is, the histologic or the under the microscope sort of pathology of osteosarcoma is essentially indistinguishable between the dog and the human. There's no one who can look at one of these tumors and say, that's a dog and that's a human. They are indistinguishable. They have the same cell types, the same patterns of growth, and the same sort of mixture of tumor types and histologic patterns. So the middle panel shows you where Jess, who's my pathologist at the NCI, went through and annotated the different areas of histologic pattern. So she used a computer program to literally draw these little regions of interest around areas where she saw hemorrhage or necrosis, which is where the HN is, or where she saw chondroblastic differentiation or evidence of giant cells. And so when a pathologist goes through and just carefully outlines these areas over, hundreds of scanned images, you can then take that data and train an AI algorithm to recognize them. And so after we did all that, the AI algorithm was able to then learn how to characterize and detect these different histologic patterns. And then when you feed it the clinical data, it can then say, well, this particular mixture of patterns is highly correlated to this specific clinical outcome. So this is an example of how we did that based on the data that the algorithm gave us. So in panel B, you see a principal component analysis that is essentially saying, how do the data group? Do they form like clusters or groups? And in this case, it looks like there are sort of 3 distinct clusters. And when you go iteratively through that and ask, well, what are the predominant features of cluster 1, 2, and 3? And so panel C shows you, again, those initials. So CB stands for chondroblastic, FB stands for fibroblastic, GC is giant cell, N is necrosis, OB is osteoblastic, and VR is vessel rich. So the algorithm says, well, I saw this and this and this in this cluster, and so on and so on. And when you iteratively sort of look at the data, panel D shows you how grouping clusters 1 and 2 together versus cluster 3 dogs all by themselves, there's a significant difference in the disease-free survival of those dogs. And then when you look statistically at hazard ratios, so these are statistical tests that basically say, let's look at a bunch of different factors. So ALP is the serum alkaline phosphatase level, age, obviously the age of the dog, sex of the dog, weight, type of treatment, and a cluster assignment. The only 2 of these that were statistically significant was the weight of the dog and what cluster it was assigned to, with the cluster 3 dogs doing significantly not as great compared to the cluster 1 or 2 dogs. So that gives us then a method for trying to apply this same type of prognostication with AI and machine learning to a scanned set of human images to see if what happens in dogs and works in dogs also works in humans. Because if we all agree collectively that the tumors look really similar under the microscope, this type of an algorithm should perform very well. So we are working with the Children's Oncology Group to get access to images from Children's Oncology Group trials to see if the algorithm that we've created and trained on dog tumors can work in similar fashion to those that are collected from humans. So stay tuned, because this is an ongoing project, and I hope to have some data later on this year to come back and share with you more about this. Okay, so the next topic is let's talk about the immune system for a minute. So canine osteosarcomas have long since been sort of described that they have immune cells, white blood cells, that traffic and crowd around the tumor within the microenvironment, sort of the neighborhood that surrounds the tumor tissue. But up until recently, that pattern didn't have sort of any clinical significance, right? So pathologists would see it and comment on it, but no one really knew, what it meant. And now that we live in an age of immunotherapy getting a lot of attention, rightfully so, the question is, maybe there's a link to clinical outcome, and our data set could be the ideal way to explore that. So what I'm showing you here is just the top 2 images are scanned H&E stained tumor tissue from a dog. And that first panel on the left shows that there's lymphocyte clusters, like aggregates in the tissue that surrounds the tumor. And again, Jess, through a lot of hard work in the lab, created a nice validated panel of markers that she could apply to the tumor tissue to call out the individual different cell types. So CD3 and CD20 are lymphocyte markers along with MUM1. CD204 and IBA1 stain macrophages. And so when you have this kind of a panel of markers up and running, it helps you then count and characterize the location of all these different immune cells. And maybe we can figure out that they have, significance for predicting outcome. So with that in mind, we had, again, another really talented postdoc in the lab a few years back, Josh Mannheimer, who took the RNA-seq data from our canine tumors and asked some questions about whether or not the transcriptional profile of these tumors could be associated with the outcome of the dogs that were on that, again, that big trial that we created the data set from. And what he did was actually look at some published data that other groups had created gene signatures that were noted to be present in dogs with osteosarcoma, but it wasn't really known whether they had any link to clinical data. And so again, our data set really helped us open the door to that. And it turns out that when you apply a gene signature that has a lot of immune response associated genes, you separate dog tumors into patient groups that have good and bad outcomes. And so that really tells you that the more active or enriched the immune system is and the microenvironment around the tumor, the more immune cells that are in there, the better these dogs would do. And when we applied that same canine-derived signature to human patients, it similarly segregated those patients into the two groups that are shown here. So the Kaplan-Meier curve, which is on the bottom right-hand side of the screen, is derived from patients that were in the NCI Target initiative. And these were patients that did not have metastases when they were diagnosed with osteosarcoma. And the orange group and the green group are significantly different than each other. And the segregation of those two groups of human patients was created based on a dog-derived signature. So again, just gives us a lot more information about feeling secure in these statements we make about dogs and humans having a lot of shared similarities. So to take that a step further, again, back to Sushant from the AI team, he was interested in continuing to work on this data set from and really used Josh's work as an inspiration to take it a little further and ask questions about this immune microenvironmental signature and whether or not he could use AI and machine learning to do additional interrogation of the data. So what he did here, and so this is a little complicated, so I'm going to walk you through it. The little sort of graphical abstract on the left just shows you how when you get gene expression profiling data, you can actually use something called deconvolution software. And that software says, okay, well, I see all these different transcripts that theoretically indicate that there's a specific number of immune cells that created those transcripts. So it sort of goes back in time to say, well, if the genes are expressed here, that means that I'm able to estimate abundances of all these different cell types that are responsible for that output. So that method of deconvolution then allows you to cluster samples based on estimated cell abundances. And so when you do all this fancy math, essentially, which is really impressive to me, not being all that strong in math, you create not just two groups, but three. And so at the top, there's an immune enriched group of dogs. There's a group at the bottom called immune depleted. And these dogs' primary tumors that are called out in the yellow didn't have nearly as much enrichment of the immune-related genes as the top group did. And then there's this middle group that has some immune cell signatures, but also has genes that are associated with vasculature and stroma and extracellular matrix components. So we call that sort of the immune-enriched ECM group. And so then when you take that same algorithm, that same method, and apply it to tumors that were harvested from dogs that went to a postmortem exam after they passed away, their owners sometimes say, well, I'm interested in learning more, so let's do a postmortem exam on the dog and figure out what else was present at the time of his passing. And when you do that, we harvested these metastases from not just the lung, which is obviously really the most common place metastases show up, but also in other areas like liver and kidney and bones and other locations. And so it's a little difficult to see, but on the right-hand side of that heat map on the right, you see that the subtype, IE, IEECM, and ID are called out in the dark purple, pink, and blue. And you can see that those subtypes are represented across all the different locations of metastases that were collected and analyzed. And what we learned here is that there's really not a strong correlation between the cluster assignment across these three groups in the primary tumor and then what their cluster assignment might be in the metastatic setting. So we have a lot more to learn here, I think, because that has, I think, implications for therapy, meaning that you wouldn't necessarily be able to expect that a therapy that you would prescribe based on the cluster assignment at diagnosis would be the same cluster assignment and potential linked therapy that you would prescribe for that patient when they, if or when they develop metastases. So again, this is sort of a developing story. So stay tuned because I think we've got a lot more to learn here.
So does that mean
So then
that,
the other thing is, oh, sorry, go ahead.
sorry, just a
Yeah.
quick question. Sorry, I'm just trying to, so does that mean then, because that first was the clusters from primary tumor, second was clustering based on the metastatic sample. So does that mean
Yes.
TME is shifting and changing from primary to metastatic disease?
It does. And I think, again, the other thing we need to remember is that all these dogs went through chemotherapy. And so what we're not totally sure is what role is the chemotherapy treatment doing to sort of shape the way the disease changes. I think it is changing it. The other thing that we are trying to work on now is understanding the landscape at the molecular level of the lung before the metastases grow there. Because I think many people you've had on this webinar and that you hear from at Factor talk a lot about that microenvironment, that sort of pre-metastatic environment that is sort of, the nest is sort of feathered, right? And it becomes this hospitable environment for metastases to grow. And we need to better understand that as well. And that will help, I think, just sort of add more clarity to this puzzle. But this is a unique finding. This was not ever described before, and it really is exciting for us to be able to have this level of understanding, and then it just allows us to have, you know, more and more questions that will help us figure out the most important thing, right, is what therapy is the most appropriate one to use in all these different settings. Thanks. That's a good question. Thank you. So the other, you know, sort of idea here is, well, all right, so now we've got three groups. What does that mean, and why should we care about that? And the reason, again, is now still in the dog, we have this finding that's very consistent with what Josh had found in the first two group analysis, that the immune enriched tumors are associated with a much better outcome. And that makes sense, right? So the more aware the immune system is, maybe the better the outcome will be, as opposed to the tumors that really looked like they were being ignored by the immune system. The immune depleted patients did not fare as well. And so then the question is, you know, what are the individual cell types that might be relevant here? Because this really then speaks to therapy. What are the cell types that are most highly associated with the best and worst outcomes? Because what we want to do, obviously, is shift everybody up. We want everybody to become more immune enriched. So what are the barriers to that? And so you can see that the shorter survival is associated with ECM, which is extracellular matrix, or stromal components, and vasculature. And one of the things we're wondering about is whether or not that extracellular matrix and stroma is acting like a physical barrier, like literally preventing the immune cells from getting into where they need to be. And what are some ways that we could battle that? So, you know, this was, again, a really exciting moment to say, okay, now we're really onto something that we might be able to target. So then the next natural question is, all right, well, so that's great for dogs, but what about people? So when you look at publicly available data sets from humans, like Target and those that have been deposited in GEO, the Gene Expression Omnibus, that have clinical outcome data attached to them. So we're after, like, not just looking at transcriptional typing, but we want to know what it means to the patients. And when you look at the method that we developed in the dog, it similarly identifies the presence of those three conserved TME subtypes. So the immune enriched are the minority, and then you've got these two other groups that, depending on the sample size, comprise the majority of the patients, consistently present in dogs and in humans. And then taking it a step further, this algorithm also predicts outcome in people. So again, putting all this data together, 160 primary tumors from dogs or from humans, you get that same sort of breakdown. The minority are the immune enriched, and then you've got these two other groups. And then there is a significant association with outcome based on that transcriptional profile that was, again, derived from dogs. And in the non-metastatic target patients specifically, these 53 patients that we were able to look at their highly granular clinical data, the prediction of metastases developing within 5 years of diagnosis is highly associated with the cluster assignment. So that's depicted in that green, red, pink box on the bottom right. So this gives us, I think, a lot of things to work from, a lot of really new information, a really exciting time for us to build upon that. So that's what we're working on. Another part of this, so just to go back to the postmortem exam of dogs. So this is not a common thing in dogs, but it's common enough that when we worked in a really large patient population across veterinary schools that enrolled on that trial across the US and Canada, we actually had 72 dogs that went through a postmortem examination where we could really confirm where exactly all their metastases were. And it turns out there's a group of dogs that never developed lung mets, and overall had a lot fewer metastases. And remember that these were all dogs that were treated the same. They were all from the same clinical trial. So why, right? What was so different about this admittedly small cluster of dogs that could help us understand better, again, that the lung is our prime target here. We really want to understand what protected these dogs from getting lung metastases. Now, they still got metastases, and you can see it's in the bone, the second most common location. And I show you the overall survival and disease-free interval curves below there, and they overlap. They're not statistically different from each other, the cluster 1 versus cluster 2 outcomes here. And that's because these dogs were euthanized. Their owners felt that, it was time to let them pass. And so in that setting, the survival is not a reflection of their disease burden. It's really of their owner's wishes. And so we always have to just keep in mind that probably the best parameter that we can use in the dogs that's transferable to humans is either progression-free survival or disease-free interval. Those are sort of the same thing. So we want to dive more deeply into cluster 1 to understand why are they different like this. And so it turns out they have different transcriptional programs. So this is gene set enrichment analysis, which is a statistical assessment of pathways that are associated with different genes that are up or down regulated. And they point to sort of global pathways within the cell. And so the pathways that are enriched in cluster 1 have a lot to do with interferon alpha response, versus the ones that are enriched in cluster 2 are more associated with like cell cycle and MYC and KRAS signaling. There is a little bit of immune inflammatory response and allograft rejection sort of sprinkled in there, but they look really different, I think, and that's maybe the point. On the right-hand side is really just another way to look at that data by pathway. So panel B, you have cluster 1 is in the pink at the very top of that heat map. Those are the small number of cluster 1 dogs, and you can sort of go pathway by pathway and look at each individual gene that's curated in that pathway to see how it's different or the same between these two clusters of dogs. And so this kind of data really helps you dig into, what's going on here. And are there targetable pathways that could be, leveraged therapeutically, but also I think more importantly, help us just understand this biology better and really clearly show us that there are some subtypes in dogs that behave quite differently, even when they receive the same therapy. Okay, so moving forward, what else are we working on? So it's a lot. I think a lot of what I've shown you today has really helped us realize that we need to do some things that relate to the spatial arrangement of these different cell types that we're seeing. So maybe you guys have heard about spatial profiling or spatial transcriptomics. And essentially all that is really asking in the tumor tissue, in these different like neighborhoods and locations of these cells, what are they doing? Specifically, and where are they located in proximity to the tumor and to each other? Are these immune cells talking to each other, or are they separated? Are they physically excluded from the primary tumor, so they really just can't get in there to do their job? And so this is where we're really working hard to understand that level of detail better. The other thing that we're doing is looking at the DNA side. So mostly what I showed you today is RNA data, expression data, but we need to also go back to the DNA side and ask about mutations and copy number gains and losses in individual genes or chromosomal segments, because I think osteosarcoma is a pretty chaotic disease. The genomes are often really highly rearranged, and it makes it really hard for us to sort of make sense of that sometimes. And I've definitely learned that from listening to the physicians and basic researchers present their work. So we really thought that would be an important part of our work as well in the dog. So stay tuned, because there's more coming on that topic. And I think all in all, I mean, our goal here is to really just, again, create a better understanding of this disease that we're trying to treat in dogs, and integrate that data with any and all human data we can get our hands on, and really understand where there are similarities and differences that ideally lead to prognostic biomarkers, new druggable targets that we can then share with the community, because that's really important for us to get the data out into the community. And then either our lab or others, or in collaboration with each other, we can take some of these ideas back to the lab and really validate them in mouse models. And then when we have sort of our top, draft picks, we can then come up with the next generation of clinical trials in dogs that can hopefully then provide new ideas for human trials. And then again, it just, because we collect more tumor specimens, we repopulate the biospecimen repository, and this just turns into sort of a self-perpetuating wheel of knowledge. That's what I'm hoping for. Clinical trial updates. So thank you again, Christina, for all of the information and the creation of the canine trial page. That is like really amazing and wonderful. I wanted to also let you guys know that we work in other disease types too. Most of what we study from the clinical trials standpoint is osteosarcoma, and we have several completed and also open enrolling osteo trials that are listed here. As you can see from the list, this is heavy immunotherapy these days, and from a lot of different aspects. So vaccines and cytokines and small molecules, antibodies, antiviral immunity is being repurposed for solid tumor management. So just a lot of exciting stuff. And we wouldn't be able to do any of this work without the cooperation and participation of the super talented, smart veterinary oncologists that work across the country and in Canada to help us enroll dogs on these trials. So they are a huge part of what we do, and super, super valuable to everyone's, the ongoing knowledge. So I think, where are we going? So it's definitely always through the comparative lens. We want to understand the disease and show the similarities and differences that we find to the community, and get the data out for everyone to look at. Really important, I think, for us to keep talking with our physician colleagues about what we're learning and how we can do better, how we can do things differently to be as impactful as possible. This whole sort of lung microenvironment, I think, is really interesting, and we want to understand that better, since we certainly see a population of dogs that seem to be protected from that. What makes them different? And then I think just, again, keep the conversation and the questions and the ideas flowing, because it's just so important to making sure that we are as impactful as possible with our work. So into that, on that realm, the Integrated Canine Data Commons is something that the NCI set up about 5 years ago. And it's an open source repository for data that sits within the Cancer Research Data Commons sort of cloud-based repository of human data. And it has a lot of cool like tools and workflows in the cloud that can help people who are interested in accessing and integrating data work in that space and come up with some cool analyses. So again, this is how you find it. There's a great help desk too, and some tutorials on here that can help you if you're really interested in either using your own tools to download data and work on your own local sort of pipelines. But there's also cloud-based tools and pipelines and methods that are available through the CRDC network that can help you if you don't actually have that expertise in your group or in your lab. So this is, I think, a really important thing for researchers across the world, around the world, to really get a better appreciation of what's available in dogs. And this isn't just osteosarcoma data. There's all kinds of stuff. And everything I've shown you today is actually in here already. So anybody who's interested in accessing it and working with it, we would really love to hear about it. So please use it, and please tell us what you think. So anyway, this is just the user interface. So it gives you an idea of what types of breeds and what types of diseases are present and what kind of data you can access. So these are all the people who helped me along the way. Certainly, I am so happy and proud to have all these folks with me in my group, and I'm so proud to be able to show the work that we have accomplished so far, and excited to tell you in the future what else we can learn and share with you. And yeah, happy to take questions if there are any. Seems like there's some good Q&A and chat action happening. So I will stop there and would love to hear what you guys think and what questions you have.
Thank you so much. I mean, so much great work. It's kind of crazy, actually, how much is going on. And I was actually thinking, because I got a sneak peek at your slides, you had that slide up front that had that really beautiful visualization of kind of all the ways that you're kind of analyzing the samples. And I was like, that would have taken me weeks just to do the create that visualization, let alone create that in real life. So anyway, thank you. Yeah, we have a couple questions so far in the Q&A. Sammy, if you want to start with those.
Yes, so we have one question from Michael Lizardo. He said, were you able to get any proteomic data from these canine patients? And do you think the surfaceome of the proteomics and the dog lung mets can find targets relevant to human patients?
Yeah, no, that's a great question. Hey, Mike. I know Mike. So we are working on the proteomic space. That's a really developing area. I think surface proteome is super important to understanding the sort of things that live on the cell surface that could be good drug targets. So Rosa Wynn in POB is a new tenure track investigator that has a real interest in CAR T development for human patients, and she's got some expertise in proteomics. So we actually sent her a bunch of samples, and I'm hoping to see some data at probably sometime this summer that we can try to learn from. So that's a great question. It's good to see you.
Okay, and then we had one other question from Alicia, and she said, have you looked into the sorry, I'm kind of butchering these words.
The neutrophils, yeah.
Neutrophils in your molecular osteosarcoma subtypes.
Yes, and so actually, if you want, I can go back to that slide and show you. Let me go back, because we can look at it specifically to answer that question for you. Okay, close your eyes so you don't get sick. Moving kind of fast here. So this is the slide. So it looks like when that cell-specific analysis was done for neutrophils, they were not significant. So we did look at it in terms of the deconvolution of the transcriptional data relating to the relative abundance of that cell type. And although they were present, it doesn't appear that their presence really negatively or positively impacted outcome in this data set, at least, but it's probably something that we'll continue to look at. And remember, this is data from primary tumors. This is not data from lung mets. So that will be something that I think would be potentially interesting in that other, in that setting.
Thank you. So I have a
Sure.
couple questions, and again, please feel free to throw any questions in the Q&A. But some questions around some of the kind of challenges, I guess, or differences between dogs and humans and how you kind of reconcile those. So, so one is that, dogs probably I imagine, get diagnosed later. I mean, because the same challenges with humans, right? No one really suspects that it's going to be a tumor, but particularly dogs, because they can't vocalize some of their symptoms, I imagine it takes them longer to get diagnosed. And so wondering if that patient group often does present with micrometastatic disease at diagnosis, or like visual, not even micro, like visible metastatic disease. And then also, just unfortunately, because of the survival, it's 10% survival rate. So when you are looking at kind of risk assessment and kind of trying to stratify, it's a little different with dogs, because unfortunately they probably all fall into that high risk bucket. And so I guess maybe what are some of the, I don't know, you actually addressed this a little bit, but maybe you can talk more about how you then kind of stratify that in canines. Let me start with I had another related one, but let's start with those two, because otherwise we'll forget what the other questions were.
No, totally get it. And it will be easier for me to answer that question if we go back to this slide for a moment, if you don't mind.
Yeah.
So let's go back to early slide here. This. So that survival curve shows you disease-free interval in days. So can you see my cursor, my arrow? Okay. So these are the dogs that can't even make it through chemotherapy without developing overt metastatic disease. And I would tell you that I think 90% of dogs that we diagnose already have microscopic metastases. And the reason I say that is because back in the late 90s, there was a study of just amputation-only osteosarcoma canine patients. So before really widespread use of chemotherapy, the question was posed, what happens if you just amputate their leg? Because we don't do, I shouldn't say we don't do a great job of limb sparing and other limb salvage techniques, but you can imagine that's really hard to do in dogs that present with a varying degree of body size and body conformation. And just, I know that's not just one surgery, it's like 7 surgeries. And dogs are just maybe not the best patients to sit and let themselves heal. And also we want them to be dogs. We want them to get out and enjoy themselves. And honestly, they don't really care if they're missing a leg. They just want to be pain-free and happy and play and all that. So back in the 90s, when we did this amputation only study, every dog really, with the exception of a very small minority, developed METs, which tells you that METs were already there when the leg was removed. So the horse was sort of out of the barn, so to speak, no veterinary pun, So one thing that I'm working on actually with Rob Rabune, who's a veterinary oncologist and a funded osteo researcher out at the University of California, Davis, is an updated natural history study in dogs. So because that data is from like the 90s, we thought this is an important thing that we need to learn more about. We need to understand the biology of the dogs that just have an amputation and no chemotherapy, because to be truthful, a lot of pet owners don't want to give their dog chemotherapy. And when you show them this curve and say, this is what you can expect, a lot of people are like, why? Why would I put my dog through chemo? I don't want them to have risks of side effects and feel sick. And maybe they don't have the financial means to do it because, again, we're relying on a lot of people to have disposable income to treat their pets for cancer. So we're really interested in what's up with this group of dogs who really can't even make it through chemo. It's sort of like the chemo is not even active. It's not doing anything for these dogs. But then similarly, like, who are these dogs out here that are living 2-3 years longer? Why? They all got the same therapy. I think that, yes, do they all get 90% of them? Do they get metastases? Yes. But there's a huge difference in time between this group up here and this group out here. And I think it, behooves us to understand that better, because right now I don't have any way. Well, now I do, because I have transcriptional profiling, but out in the clinic, there's no way to tell those dogs apart. They look identical. There's nothing about them that looks different from the, just the clinical management side. So I don't know if that answered your question, but I can answer the other one too, maybe, if you can remember what the other question
Yes.
was. I don't remember.
Yeah. Well, actually, so this is, and you touched on this too about chemo and how chemo can affect the TME. But I mean, it's kind of funny because I know, especially even with mice, oftentimes we're like, oh, well, why didn't you test the mice with, MAP? And it's like, well, mice can't tolerate MAP. Like, we can't.
Yeah.
And so same thing with dogs. Like, I know dogs have a different chemo regimen, right, than humans. And so I'm
Yeah.
also wondering how that might impact our understanding. Because as you've said, like, we don't know if chemo might be
Yeah.
changing the TME.
Well, and so that's a great question. And I would tell you that we tried for decades to give dogs MAP in various flavors. We really did. We tried to give them sequential or combined MAP agents. Methotrexate, let me just say, is like really hard to give dogs from a tolerability standpoint, but dogs can tolerate doxorubicin and platinum agents fairly well with good supportive measures and appropriate dosing. But what we learned, I think, and I'm condensing like 30 years of research down to a single sentence, is what we learned is that you don't actually do better by them by doing that. You make them sick and you make them miserable, but you don't actually, they don't live longer. And I would tell you that I think, and I would open to the floor for comment. I think that dogs represent the 30% of human patients that are not served well by frontline therapy. I think that's where dogs are sitting. I think, are there a handful of dogs who are going to do well no matter what? Yes. But I think by and large, the dogs are representing that really underserved patient population. And I think that's also echoed in other diseases. I think dogs with prostate cancer represent the worst of prostate cancer in men. I think dogs represent the worst of melanoma in humans. So I think the more that we as a community do this kind of work, the more we realize that there are echoes that are really throughout different types of cancer. And there's a lot of biologic convergence. So I say that because dogs and humans are getting a lot of the same kinds of cancers, but they're not getting it for the same reasons. So, dogs might get lung cancer, but they're not getting it because they smoke, right? So I just wonder a lot about that, because I think some of the upstream events, like the inciting events, are different, but then the diseases really look a lot alike. So we have a lot to learn there. And I think it just sort of speaks to, there's no shortage of questions that we all have. So it makes it scientifically exciting to think about what we can learn.
And to that point, I mean, I think there have been already studies that have shown that there are similar mutations in canine and human osteosarcoma. And is the incidence rate kind of similar to, like, for some of the kind of common ones like P53 or RB1, ATRX?
They are somewhat similar. And actually, I should have put that slide in here. I didn't want to dwell too much on like granular mutations and have everyone fall asleep. But yeah, I mean, I think the hallmark events we see, P53, RB, the cyclins, MYC, like we see those. And I think in most cases, they are sort of occurring at similar frequencies. But what's really interesting is that the actual like genomic lesion between, and P53 is a good example, where in people that's mostly like point mutations, but in dogs it's different. It's like a chromosomal loss. So at the end of the day, is P53 still abnormal? Yes, but the way it's happening is different. And again, that sort of just speaks to this like convergence thing that I think I see a lot of evidence for that. Now, having said that, there's some things that are different in the dogs. Like one good example is that dogs get, or seem to, at least based on the data we have, a much higher incidence of mutation or loss in something called SETD2, which is an epigenetic associated pathway gene. And so we're really interested in that too, because so first of all, why? And then second, does that represent any sort of recapitulation of this high incidence of metastasis? Can you link that prognostically? So I think that's where a lot of the developing work we have on the DNA side, like the mutation and copy number side, that actually I'm planning to have a more mature story to show at Factor this year. So my plan is actually to put that that piece of the puzzle in there and then really show you, I think that we have evidence for that as well, that there are some mutations that appear to have some value in terms of predicting outcome, whereas I don't think that's really been shown on the physician side at this point. So yeah, stay tuned.
So we do have one comment from Jamie. She said, "Thank you for saying that, Amy. We have been beating the same drum of different beginnings and a similar end for a while, but I think when you say it, people will really pay attention."
Oh, that's Jaime Modiano. Hello, thank you. Good to see you.
Yeah, and then I had a couple questions. They're maybe not as like intricate and smart sounding as the questions Christina was asking, but I think you already touched over. One of my questions was if the type of chemotherapy drugs used for humans is the same that are used for dogs, but I know that you just said that it's a little bit different because like methotrexate isn't good for them. So then to go off of that, like what is the chemo regimen for dogs? Like you were saying that how humans have maps, like is it just different based on like the progression of osteosarcoma, or how do you guys go about treatment?
I think a lot of the veterinarians that are treating osteosarcoma, I think if you put a bunch of us in a room, we'd all like argue with each other about it. But I think the common theme is that most of us agree that a platinum drug is like the key. So dogs in this country, at least, are mostly going to get carboplatin as a single agent, and anywhere from 4 to 6 treatments. The old school cisplatin treatment in dogs, again, because of that drug being so damaging to kidneys, we don't give that drug in a way that protects dogs' kidneys. So it used to be that's all we had, and it wasn't really a question of if they would go into kidney failure, but when. And then carboplatin came along, like I'm guessing it was like probably the early 90s. And we were like, yay, because that's much more kidney friendly. And then people were like, okay, well, maybe we should do, again, in that sort of time where people were trying to recapitulate MAP. Well, maybe we could do like carboplatin and doxorubicin together, either separated or like one after the other, or like all carbo and then all dox. And people like there, you can count like a stack of papers like this that where people were trying to like sort of fine tune that. And I think in the final analysis, there's some really good papers that people wrote where they went back and like just pulled all the data and said, what really is the key here? And I think all of us sort of collectively agree that it's the platinum drugs that are the most biologically active in the dog. And so these days, I think we've all sort of like waved the flag and been like, all right, it's platinum. And the clinical trial that I showed you, that curve, those dogs got an amputation and then 4 doses of carboplatin.
Okay. And then I'm kind of glad you brought up the amputation because that kind of leads into like my second question, I guess. Like I was wondering what the survival rate is for dogs with osteosarcoma, because obviously it's hard to diagnose, and by the time you diagnose, it's really hard to treat. And then I guess my next question that kind of leads into that is if a dog like beats cancer per se, like with amputation and with chemo, if it reoccurs, is their survival rate lower too? Like it's usually, or is that usually not common? Like they don't make it?
So let me answer your first question. So overall, if it was your dog and I was talking to you as a pet owner, I would tell you that if you went through the sort of like standard amputation and chemotherapy thing, that the average survival is about 6 months, which is not great, right? And it used to be that we thought it was more than that we thought it was more like 8, 10, 12 months, but our data suggests it's more like 6 months. 6 to 7 months. And that's really like, I think all of us sort of wondered about that, but it really wasn't until we created this really big data set and shared it with people that we realized like, we're really not doing as well as we thought we were. Having said that, I mean, there are dogs that live a long time, and you think, okay, well, now I think we have a better understanding of why, because it's likely that their immune system is just more readily seeing their tumors, right?
Mhm.
But when they do get metastases, it's really hard to treat those dogs. When they get visible metastases in their lungs, they tend to be really treatment resistant. Although there are some really talented, cool people working on, like Dan Reagan and Cheryl London and Steve Dow, like all those people you've probably heard from or will hear from. There are some really interesting treatment options being tested in dogs in that setting that I think are hugely valuable, not just from the clinical management, but also just from like just understanding the biology of metastatic disease and I think it's really different than what's happening in the primary tumor. At least it looks like in our data that really is becoming kind of obvious to us. So it's not a one size fits all situation at all. Yeah. And I think the other thing about dogs with osteo, I think some of the dogs that are long-term survivors will often get other tumors. So I think that might speak to some breeds of dogs being somewhat cancer prone. So not uncommon. Like I've seen in my career several dogs that, sort of beat their osteo and then they got like lymphoma. Like, really? Like, hasn't this dog been through enough? So that's always a challenge too, because I think dogs in general just get a lot of osteo and they get a lot of cancer in general. Their incidence is, probably at least 10,000. I would say that's a gross underestimate of how many dogs a year get osteo in this country. It's a lot. It's a big problem for veterinarians.
Yeah. Well, thank you for answering my question. I appreciate it. See, I'm a dog lover, so it makes me really sad to think about that dogs go through all this too, but it's also really nice to know that there's so many people that care enough to like research and try to make it better for dogs as well as
Yeah.
people.
Yeah. Well, I mean, you summed it up perfectly. That is what we're about. So we want to help everybody if we can.
Yeah, my dog, don't take it personally, but he just snoozed through your entire presentation. But he
Well,
did
that's not the first time that's
make sure
happened.
listen if you
That is
want it.
definitely, that's happened to me before. So I'm not offended.
Yeah, I just checked on him. Yeah, he's out. Well, thank you
He's
so
out.
much. Thank you so much, Dr.
Of
LeBlanc,
course.
for joining us on OsteoBites today and for making it better, both for our canine friends who really are our best friends, both getting us through life and also helping us kind of solve this whole cancer puzzle and making it better also for our pediatric osteosarcoma patients. More information on this and all OsteoBites can be found on our MIB Agents YouTube channel and on our website at mibagents.org and at your favorite podcast place. And next Thursday on May 16th, we are dropping a new episode of our AYA podcast, Osteo, where Osteo Warriors Camille and Mia spill the tea on living with cancer as an AYA. And Sammy is actually a special guest on the episode that's dropping next week. They share their personal experiences with long-term side effects from treatment from osteosarcoma. So again, you can find the episode on our website and on your favorite podcast place, and that's dropping next week. And then we're going to be back the following week with a special episode of OsteoBites. We're going to be joined by Dr. Matteo Trucco, who's a pediatric oncologist at Cleveland Clinic Children's, and he's also the chair of our MIB Agents Scientific Advisory Board. And he's going to provide a preview of our Factor 2024 scientific panels in layperson terms. So we'll provide a brief overview of some vocabulary and terms as a helpful resource for patients and caregivers attending our conference and interested in the scientific sessions. You can find our OsteoBites lineup for the next few months on our website. And if you have any ideas for future topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again so much, Dr. LeBlanc and Sammy, for joining us today. And thanks to all of you. And we hope to see you back here in a couple of weeks when we talk to Dr. Matteo Trucco about our Factor sessions. Thank you so much, everyone. Thanks, Dr. LeBlanc. Thanks, Sammy.